And this will be in 3 parts. It will be first on replication of DNA, and then it will be on transcription from DNA to RNA, and then it will be on translation from RNA to proteins. So, but first of all, are there questions on the stuff that we talked about yesterday? In such a case, I'm very happy to try to answer your questions. Or if you have questions that you would like to ask to me more privately, this is my email address, so you can ask questions. And if you come to think of something later, then just ask me. The central dogma, as I said, DNA needs to be replicated. It makes copies of itself, and then this DNA is then copied to RNA, to messenger RNA or other types of RNA, and this is called transcription. But there is also a reverse transcription from RNA back to DNA. Say, for instance, the HIV virus has such a mechanism. But then from the messenger RNA, we go through the translation process to proteins. So this will be the first hour, this will be the second hour, and this will be the third hour. And I think we need a break somewhere in the middle So, first of all, the packaging of DNA. How can a genome of 3 billion base pairs be packaged into a small cell of 1 micrometer? So the genome would be roughly 1 meter long, the human genome. So a cell which is 1 micrometer, how can that contain the whole thing? Of course, you also know that the human genome is not the biggest genome of all species. I think there are very primitive organisms having much bigger genomes than what we have. Well, the DNA must be compacted somehow. And in eukaryotes, histones are associated with the DNA. So the histones is one important part of the packaging. So here is, for instance, the double-stranded DNA, and the size of this is roughly 2 nanometer or 20 angstrom. Then we come to the nucleosomes, and the size of a nucleosome is roughly 110 angstrom or 11 nanometer. Then we come into a fiber which has a 30 nanometer dimension. Then the chromatin makes loops, and these are in the size of roughly 300 nanometer. And then these can condense in different ways, so this becomes the size of 700 nanometer. And then the chromosome has the dimensions of roughly 1400 nanometer. So there's different levels of compacting the whole thing. And I think we have a good idea how this looks and a partial idea how this looks, and I'll show you how that is done. So the histones, there are 4 main types of histones: H2A, H2B, H3, and H4. They form heterodimers. So H2A and H2B, they form dimers, heterodimers. And then H3 and H4, they form also dimers, but these H3, H4, they then aggregate into tetramers. So one nucleosome contains 8 histones. 2 of each forms 2 dimers. So there will be two of each of these 4. And in addition, there are also histones called H1 and H5, which are found between the nucleosomes. And I'll show you how these are organized. So there is a standard structure of the histones. So all H2A, H2B, H3, and H4 are related. And you see they have some similarity in the structure. These are alpha helices here shown. So there is a long alpha helix, and then there are 2 short ones on the sides, and then there is some variation between the different classes. And this is called the histone fold. this part of the whole thing. And then there is a long N-terminal, amino-terminal tail, and then there's a small C-terminal part also. And actually, the histones are the most conserved proteins that we have between species. They are, say, doing the exactly same job in all types of organisms. And the amino acid sequences here have been perfected for this type of job. So there's very little variation between, say, human and spinach or carrot, if you like. So this is the structure of the histone fold. So this is the N-terminal helix. Here's the long helix. And then there's a C-terminal helix. And if you look at it, it looks as if it has a twofold symmetry. But of course, this is not a real twofold symmetry because the chain has a certain direction. Amino end is here. Carboxyl end is there. So it's just a virtual type of symmetry. So one is calling this type of fold for helix, turn, helix, helix, turn, helix, this type of motif. And it has a twofold repeat. So here's then a histone dimer. So here we see two of the histones, maybe H2A and H2B. So you see the two long helices here, they pack against each other, and then the short helices are top and bottom. And there's also a small piece of beta structure here between two short pieces up there and also up there, beta bridge it's called, connecting, making contacts between the two monomers. So here's then the organization of the nucleosome. H3 and H4 form the dimer, and subsequently two dimers form the tetramer, and then to this is added two dimers of H2A, H2B to be forming the total core of the nucleosome. And compared to viruses, which I won't be talking about, here in the nucleosome, the protein is in the center, and the nucleic acid, the DNA in this case, is found on the outside. Well, what is this? This is the extra histone, in this case H5, but H1 would also be sitting here. So the DNA here makes almost two full turns around the nucleosome. So each nucleosome has two turns of nucleic acid, and it's roughly 150 to 160 base pairs contained in one of those. Now, this is then a simplified drawing of the nucleosome. You see the four histones in the middle here, forming the core of the whole thing. You see two turns of DNA, and here's the extra histone H1 or H5 that I showed you before. And then this is organized into some way into the 30 nanometer fiber particle here. And this forms something that is called beads on a string, pearls on a necklace. But now, due to further structural studies, one has observed that four nucleosomes, they form a tetrameric arrangement. So two nucleosomes are sitting here, and two are sitting on top of each other here. So if you turn this by 25 degrees, then you see that here now, these are sitting on top of each other, here they are sitting on the side of each other. So this is then the first type of packaging into the larger structures. So first of all, the nucleosome and four nucleosomes, they form some type of aggregate. And then if you start from four, so here then in the bottom there are four, then four in purple, 4 in orange and 4 in blue. So this is the way the set of 4 nucleosomes pack to each other. And so here you see the same thing but drawn in a different way. The blue, the brown, and the purple seen in 2 different directions. So this is then 12 nucleosomes, the aggregation of 12 nucleosomes. And then you have the whole arrangement of the 30 nanometer fiber, which is continuing on this type of motif. So you go through the whole thing here. So there is then a fairly increasing understanding of the aggregation of the nucleosomes and packaging of the DNA into the cell. Now, when you want to replicate the DNA, then this packaging is a severe problem. You need to open it up because otherwise you cannot make the copy. So you need somehow to remove everything. And this is then a summary description of how this is done. Before cell division, the genome needs to be duplicated, of course, and there are a whole range of different proteins and enzymes which have to work on this. So, for instance, you need to, first of all, remove the histones, and I can come back to that in the next hour when we come to transcription, but let's leave that for the moment. But the double helix of DNA needs to be split, and this is done by a protein called the helicase. And when you open up the double-stranded DNA, you need to protect the single-stranded DNA from degradation. So certain single-strand binding proteins are sticking to the DNA to prevent it from degradation, because that would be terrible if the DNA is starting to be broken down. Then you have over here an enzyme called Topoisomerases, because if you now start to pull the 2 strands apart, then you will generate super winding of the DNA beyond. And this enzyme then is cleaving one of the strands and letting it spin and then closing it again. Or in some cases, the topoisomerase will cleave both strands and let the whole thing pass through each other, and I'll show you in a moment how that is done, and then reconnecting the strands. But now, when we do the copying, the replication of the DNA, there are 2 strands that needs to be copied. And since we're moving from the 3' end towards the 5' end, then in both systems, it will be easy on the so-called leading strand, because here then the DNA polymerase, Pol delta, can just proceed along here, and while the helicase and topoisomerase are moving ahead here, this enzyme just makes a copy. But on the other side here, the lagging strand is not so easy because it's moving in the opposite direction. So it makes a new DNA strand from part, but then it has to start again and to make another part, and it has to start over and over again. So this will become then a whole set of fragments of DNA, and these are called the Okazaki fragments, which will be then ligated, connected to each other. So some proteins which are needed for replication. First of all, you need to recognize the origin where the replication should begin. You cannot start in random places. So origin recognition, and in eukaryotes it's called ORC. Then to remove the histones and deal with the histones, you have histone chaperones. And there are many of those in eukaryotes. There are none, to my knowledge, in E. coli. Then we have the helicase, and there are different ones in E. coli and eukaryotes. We'll talk a little bit about them. And helicase loader, because the helicase cannot just start to operate on its own. It has to be loaded onto the DNA. And then we have the single-stranded binding proteins, and then we make a small primer of RNA for the synthesis of DNA. Then we have something which is called the clamp loader. So this is a clamp which holds on the DNA synthase so it doesn't fall off, because that would also again be terrible. It should be always sticking to the job it's doing. So sliding clamp and clamp loader. So we have both the helicase and helicase loader. We have the clamp, sliding clamp and clamp loader. And then finally we have the DNA polymerase. So this is some parts of the proteins which are engaged in replication. So here we see one of the histone chaperones, which remove histones ahead of the replication fork and supply both new strands of DNA with new histones. Because once the DNA polymerase has made DNA, we again need to package the DNA with histones and make nucleosomes and so on. And there are many of these histone chaperones. This is one of them called ASF1. And here we see the histone dimer of H3 and H4. And this histone chaperone binds to the surface of H3, where it is interacting with the neighbor H3, because, as I said, H3, H4 makes a tetramer, so this one binds to the surface where the other dimer should be sitting. So then it can, say, remove this from the histone and hold on to it until it's needed again. And here is now a structural understanding of the complex that is involved with the recognition of the origin of replication. So here's a piece of DNA, in this case roughly 72 base pairs long, and then a complex set of proteins which are engaged in this origin recognition. So it's ORC1 to ORC6, and a protein called CDC6, and it makes a specific structure that can bend and identify the origin of replication on the DNA. So the helicases then. To copy the DNA replication, the two strands first need to be separated, and this is done by motor proteins, because certain proteins are motors. They need energy to be moving along to break up the hydrogen bonds between the base pairs requires energy. So the helicases use ATP as the energy source and move in the 3' to 5' direction along the DNA helix. And if these helicases are mutated, this will lead to severe problems. So many diseases can be due to mutations in helicases, including cancer. But now there has to be a knife in the helicase, a knife that is cutting through the hydrogen bonds between the base pairs. And what is this knife? Well, first of all, let's have a quick look at the families of helicases. You see there are at least 6 families, and 2 of them are monomers, and they contain two domains. You remember from yesterday what we talked about that proteins can contain domains. It's like, again, like beads on a string. A completed bead here is one domain, and then follows by the next domain. And these proteins, they contain two domains called Rec-A. They are closely related to the Rec-A protein. In evolution, I think this type of material, this gene sequence, has been used several times for various different purposes. So both SF1 and SF2 contain a doublet of the Rec-A. And these are involved in DNA repair, because even in DNA repair you need to open the DNA. But these other ones, these four, are hexamers, and they have, two of them have the Rec-A domain, but two others have another type of domain called AAA plus domain. And they are involved in DNA replication and transcription termination, for instance. So let's see what they look like. So here's one of the monomeric helicases called here Rec-Q, which has two Rec-A-like domains, D1 and D2. And it depends on ATP for the translocation movement along the DNA. And here's the binding of the ADP or ATP. In this case, the third phosphate, the gamma phosphate, has been removed. So in the process, I mean, ATP is then cleaved to lose the last phosphate, but then, of course, it picks up another ATP to be able to proceed. And the ATP sits between the two domains here. But down here is the active part of the whole thing, and here is a beta hairpin, which is the knife. So this purple structure here is cutting through the DNA. So here we see a complex with a piece of DNA, and here is the knife now in different color. So it's opening up the two strands, the yellow and the green strand of DNA. Then I mentioned when you're using the helicase to pull apart the strands of the DNA, then you superwind the DNA, and you need to use the topoisomerases to release this strain in the DNA. And there's a big family of topoisomerases, and I'll just mention two different cases. And one big family here is the topoisomerase 1. And it's easy to remember what 1 and 2 are doing, because 1 is just cutting one of the strands of DNA. Topoisomerase 2 cuts both. So one strand. Let it spin once or several turns, and then religate the DNA. And the phosphate at the end of the cut, that is getting covalently linked to a tyrosine, because if you let it loose, then the whole thing may get lost, and you may not be able to proceed properly, but it becomes covalently attached to a tyrosine in the protein. So here we have topoisomerase 1A, which has 4 domains, and you can see them here in different color: purple, red, yellow, and blue, and seen from 2 different directions. And the active site is here. So this is the way it happens. So here is then the first step and the linking number here is N. So it's, and then you see the DNA, and it has been opened up by some helicase here. So it binds to domain 1 and 3, basically, and then there's a cleavage of the DNA, and now the blue star that up on the orange subunit has the tyrosine, and here is now the covalent link. So then the next step here is the strand passage. So now the complete strand here is moving through, and then you religate. So the tyrosine is now losing the grip, and the two ends are religated, and then the whole thing is released. But if need be, you can go several turns around in this way. So here the linking number is N+1, but you can make several turns of this if this is needed. Topoisomerase 2 cut both strands to let one double-stranded DNA pass through another before it's religated. And also here there is a tyrosine which is involved in holding on to the cut DNA. And here's a whole family, and the different colors here along the amino acid sequence illustrate the domain arrangement. Yellow, orange, red, purple, blue, dark blue, and another purple color. So just an illustration of the general organization of this, and this then shows the complicated structure of the protein. So here it is, there's a twofold symmetry axis right through, and there are different regions which are, say, hollow, which could contain the DNA, and other regions which are locked, which needs to be opened up in proper moments. The tyrosine here, which is involved in holding on to the DNA, is sitting right there. So we'll see how the procedure is done. So there are three gates. The N gate, which is up on top. Then we have the DNA gate, which is in the middle here. And then there's the C gate at the bottom. N gate, DNA gate, and C gate. And this picture shows what happens. First, one double-stranded DNA is bound across the enzyme here in this region. And then another segment of DNA is bound into this structure up on top. Then two ATP molecules bind to the enzyme in the proper place, up on top here, and then the structure is closing around the DNA. So this now is trapped inside here. And then comes the cleavage of this double-stranded DNA, such that now this upper DNA double-stranded can be moved across the opened up DNA into this cavity here. And then in this process, the ATP is hydrolyzed to ATP and ADP. And then in the last step, this bottom structure, the C gate, opens up to release the DNA which has been passing through, and then the whole thing is religated here, reconnected. And then the last step, the final gamma phosphate is released, and you go back to, you release the DNA and go back to the, say, empty structure. And if there's a need, you can, of course, move another turn. You can go around several turns here. So So this is the way you release the strain in the DNA after the helicases have been opening up the structure. Well, what is this? This is the sliding clamp. So you see, this is a beautiful structure which has a roughly six-fold symmetry, and this is then holding the DNA inside here. This moves like a ring on the DNA. But to put a ring onto the DNA, you should need to either find the end of the DNA, which may not exist because it could be a circular DNA. So in such a case, the ring has to be opened, and this is what the clamp loader is doing. And actually, due to the existence of this clamp, the DNA synthesis is becoming much more rapid. So maybe 2 orders of magnitude improved rate of DNA synthesis. Because otherwise, the DNA polymerase may fall off. So this is holding on to the DNA polymerase to prevent things from falling off. So as I say, the clamp is loaded onto the DNA by the clamp loader. And here's a simplified picture of the clamp loader. It's a complicated structure by itself, and it breaks open the ring between 2 of the subunits, between 2 and 5 here, such that the DNA can be bound into the ring, and then the ring is closed, and the clamp loader, which uses ATP again to do this type of job. This picture shows a little bit more of the details of 3 different versions of the clamp loader structure. But we shouldn't take more time to go into that. So here is then a complicated picture of some of the enzymes that are dealing with the whole thing. So here then we have the parent strands of DNA, green one and a black one. And here's the leading strand, as I said before. So this is the DNA polymerase, this big thing here, and this is the clamp which holds onto the DNA. So they both move jointly in this direction. And here's helicase, which is opening up the DNA all along here. Then, of course, the topoisomerase also needs to be here. The single-stranded DNA here, which is not copied immediately, covered by the single-stranded binding proteins. And here's then the Okazaki fragments being produced by different DNA polymerase. It is making piece after piece after piece of then religating those. So this is then a reminder of what I showed you before, but a slightly different artistic view. Now, we come to the DNA polymerases, which do the actual synthesis of DNA replication. And this is a very large family of enzymes again. There are many different needs for different cases, and different species have slightly different ones. So here it's divided into the class A, B, C, D, X, Y, and reverse transcriptase, RT. And E. coli doesn't have so many, pol 1, pol 2, pol 3, and DIN-B. But in mammalians, we have a number of DNA polymerases, pol gamma, pol alpha, pol delta, pol epsilon, pol beta, and pol eta. And then there are some other ones here. So the ones we will focus on are the pol delta and pol epsilon, the main replicase, the lagging strand, and main replicase on the leading strand, 2 different enzymes. So just to give you an impression of the range of enzymes involved. So here is then the structure of DNA polymerase. And the simplified picture of the whole thing is like a hand holding onto the DNA. So the in the hand, there's a palm, there is a thumb, there are fingers, and there's a little finger. So little finger is this purple structure. The other fingers here are the blue. Palm is colored in red, and the thumb is colored in green here. So this is from two slightly different views. Here you see the DNA straight down, and here you see the DNA coming from the side here. And here's the nucleotide to be incorporated. So remember this, the DNA polymerase is constructed just like a hand, and we'll go into some more details of this. So here we see the different parts again. The palm is here in yellow, and the thumb is in red. The fingers are in blue, and I lost the little finger. And here's then some piece of DNA, and here's magnesium ions, which are very important in the active site for the incorporation of the new nucleotide. Now, if you compare different DNA polymerases with each other, they are looking quite different, but still they have the same organization like the hand. So here, the palm is colored in yellow in all four, and the fingers are colored in the same way. So you see, you find the different details in all those, but it's the general construction differs quite a bit. So if you look just at the palm part of the structure here, it's composed of 4 beta strands and 2 helices. This is the central part of the palm, and it's so-called double split beta alpha beta. So you come in through the beta strand 1, and this goes off to the fingers, but then it comes back to beta strand 2, which is parallel to number 1, and then it gets into strand number 3, goes through another helix, and comes back through number 4. So number 3 and 4 are also parallel. So this is called a double split, because 3 and 4 are split by number 1, and 1 and 2 are split by number 3. So it's so-called a double split type structure. And here you see the classes A, B, C, and X. And if you align them in this way, you can see that they are quite similar, even though they have different sequences and different details. And the important amino acid residues are found down in the carboxy terminal end of the beta strands, or beta strand 1. So here are the crucial active site residues painted in detail, shown in detail. So here is then the central part of the whole thing. So here is then the last base pair of the DNA, and here's the base in the template, which is now going to be replicated by a nucleotide that comes in here. So this is a 3-phosphate DN3-phosphate compound. The base is making the proper Watson-Crick base pairing to this, and this is now done with great fidelity. And then the 2 magnesium ions here, which are bound to carboxyl groups of the protein, they are participating in, say, making this phosphate attack the deoxyribose oxygen here, and to release then the 2 phosphates from the nucleotide. But actually, this general proposal has been challenged by recent studies from crystallography, where time-resolved studies showed that if you can resolve in a brief time, there's a third magnesium ion coming in, which seems to be equally active and important as the two other ones. So I think it's very likely that there are 3 magnesium ions which participate in the catalysis of incorporating a new base into the growing DNA. Now we come to another problem here, because when you are doing the replication here, say if you have a DNA which has 3 origins of replication shown by these 3 O's, you start at this point and you proceed in one direction very smoothly, and also here, but then you have the Okazaki fragments on the other side. So if you proceed and complete essentially the whole thing, and you ligate the various pieces to each other, there are pieces here which are too short to be copied properly by the DNA. So you'll form the incomplete ends of the telomers. So now this requires a specific type of enzyme which is called telomerase, which is completing these missing bits here. And if you have deficient system or mutations in the telomerase, then your new strands of DNA will become shorter and shorter with each new generation. And of course, this can lead to quite bad developments. So, as you may remember, there was a Nobel Prize given for just this investigation on the telomerase by a lady called Blackburn. So lack of telomerase leads to cell aging and death. In mature cells, the telomerase, the enzyme is switched off, because mature cells, they are not involved in further replication. But in cancer cells, the telomerase can be reactivated, making such cells immortal. So this is then a specific problem. And the telomerase is a stable RNA protein complex, just as many other complexes. Protein and RNA can make very interesting structures. And the RNA component here contains the template for adding the repeated sequences of the telomerase, or the telomers. And here we see the RNA component of the telomerase from ciliate, from humans, and from yeast. And yeast has a very large and complicated structure. Humans, not so complicated, and the ciliate is quite simple. And in this structure, there's something called a pseudo-knot piece of the RNA, which is a very complicated structure. It goes this way over to this end, then comes back and makes, so it's basically becomes, in most places, a triple-stranded arrangement. So this is called a pseudo-knot. And the telomerase protein called TERT has a number of different domains and regions here. But basically, it's arranged in the same way as the DNA polymerases. It's got a palm, it's got a thumb, with fingers, and then it has a separate domain in the beginning here. So this is the way it looks, and it's dealing with the DNA and making use of its own RNA as the correct template for the missing piece of DNA. So here the RNA is in purple. And now here the DNA is being completed. And also here the REC-A uses ATP for its function. And the monomers of REC-A, they form six-fold helical structures. So six-fold helical. It's not a circular six-fold, but it's a helical structure. I'll show you soon how it looks. And this is a monomer of the REC-A, with the ATP binding site down here. And the amino terminal and carboxyl terminal domains bind the DNA and form the oligomers. So this is the eukaryotic, and this is bacterial version. So now, the first thing that happens after the break of a double-stranded DNA, and one strand is digested away. And this, so you have now a double-stranded piece, but then you have a single-stranded piece of DNA. And now, of course, this needs to be covered as soon as possible with single-stranded DNA binding protein, these little yellow dots. And now, in the next step, REC-A is replacing the single-stranded binding proteins on the single-stranded DNA here. And this is catalyzed by something called the mediator, this protein that removes the single-stranded binding proteins and sticking in the REC-A here. Okay, so far so good. And this then forms the presynaptic filament. So here you now have double-stranded DNA and then the single-stranded DNA with a whole chain of REC-A proteins bound to it. But if this is continuous or if it is, say, segments, is not known. And then comes another double-stranded DNA from the father, possibly. And then this forms then heteroduplex DNA. So the pink strand is now making heteroduplex with the light blue instead of initially there was the red and the pink belonging together. So the REC-A protein is, say, assisting the combination in this way. And then the REC-A disappears from the whole thing, and DNA repair synthesis completes what is missing. Okay, so I think I described that. But now let's look at the structures of these things. So here is a whole set of REC-A. REC-A1, REC-A2, REC-A3, REC-A4, REC-A5, and REC-A6. So this is the helical arrangement of REC-A molecules which associate with each other. And here we see in red the single-stranded DNA bound to these molecules. And actually, I'm very jealous to the people who did this experiment because they made, say, an oligomer, hexamer of REC-A proteins by ligating one to the other. So instead of having monomers, they made a long protein which contained 6 monomers, and there was the linker between them allowed them to be flexible and find the proper way to interact with the DNA and with each other. So, and then they could crystallize the whole thing and determine the structure of both the whole complex with the DNA. So this is a very elegant set of experiments. Okay, so this is the way the hexamer of Rec A is organized with the DNA. And each Rec A molecule binds 3 nucleotides with a gap to the next. So here you see the 3 yellow nucleotides bound to the green domain. I suppose this would be here. And then there's a gap up to the next nucleotide, a fairly large gap, and also there's a gap which is not shown in this picture down to the next domain before, lower down. And here is now the situation when a new strand, say a father strand, is coming in to be making a heteroduplex DNA organized onto the single-stranded DNA here. And we said that ATP is needed for this process, and the gamma phosphate here of the ATP, the last phosphate, is sitting between the domains, between the Rec A molecules. And when ATP is hydrolyzed, it will fall off from the DNA and cause a structural rearrangement. So here we see the heteroduplex DNA when bound to the Rec A molecules. And you can see there are 3, 3, 3, and 3 organized in, say, a way which is not the normal organization of the DNA, but to do this type of careful recombination, this is needed. But now, there are some further, the whole patch, large patch of positive charges here. And of course, positive charges there would be expected to bind something negatively charged. And DNA is negatively charged. So here is then the missing information about where the other strand of DNA would be bound, but it has not been observed directly yet. So here you see the positively charged bits on the structure here, which would be very inviting to bind the different strand of DNA. So I think this was the first hour. And do you want to take your break now, or should we take the break later?